So it is my great pleasure to introduce Professor Kelsey Martin to finish our symposium. Kelsey is, as you heard before, currently the Dean of the Geffen School of Medicine at UCLA, but she's really a quintessential scientist. She received her undergraduate from Harvard University in English and American language and literature. She then served on the Peace Corps and volunteered in Congo. And then she served again here as a postdoctoral fellow in Eric Kandel's lab before joining in 1999 UCLA as an assistant professor. And what a change that was. I remember this was the year that I started as a graduate student, and it just changed things. Kelsey's lab was like a home. We all hang out there. It created a horizontality at UCLA between students that was amazing. I learned about Harry Potter. I don't know if you remember from the books, and we all read together with Kelsey's kids. And anyway, Kelsey then did pioneering work that started here with Eric on the molecular and cellular basis of memory. And she has elegantly combined electrophysiological, molecular, and cellular approaches to gain key insights into fundamental problems into the biology of memory. And I mean, related to, not as a coincidence to the previous talk, she has discovered mechanisms underlying specific changes in the strength of individual synapses and how these changes can be sustained and eventually sustain persistent memories and requiring the synthesis of new proteins. I mean, it's really amazing because the neuron as a cell is the most fascinating thing that one can think. I mean, it's always transcribing things, needs to transport it everywhere. So it's really amazing, and I think we need to go back to study more of that. Kelsey has received many honors and awards, including the membership in the American Academy of Arts and Sciences, National Academy of Medicine, and she has been also a role model for all of us, as you saw today in the panel. Please join me in welcoming Kelsey. Thank you, Kelsey. Thank you.
Well, thank you. It's such a pleasure to have Rui introduce me, because I do remember Rui in Inbal many, many years ago when I started at UCLA, and it's really an honor to be here. I'm humbled to speak in this incredible symposium with incredible talks. Columbia meant a lot to me. I work on experience-dependent plasticity, and it definitely shaped my brain and my whole personality. I think that I have had so many mentors and friends here. Eric, you mean a great deal to me. You always will mean a great deal to me. I feel like, Richard, you kind of taught me how to not worry about being intimidated by you, a very valuable skill that I try to keep up. And I do feel this incredible sadness to not have Tom Jessel here to really realize this celebration of something that he gave so generously to this community to really build such an amazing building, campus. I think about, when I think about Tom, I think about the introductions that he gave to people. And the way in which he so deeply appreciated the science and the contributions of every individual scientist. And I think that that's sort of a fitting tribute to the work that goes on here, and I'm sure will go on for years to come. So my lab is interested in understanding how experience changes connections in the brain. And we think about this very much as cell biologists. So we think about the question of how does experience change the connections between nerve cells in the brain. And we take as a starting point, and I almost feel like I should be, I think it's McGraw-Hill now is your publisher. I feel like they've gotten a lot of support today from Kandel, Schwartz et al., the sort of Bible of neuroscience. But I take as a starting point the finding that long-lasting changes in connectivity, in plasticity, such as those that underlie long-term memory, require new gene expression. And also, long-term plasticity can be synapse-specific. So when you think about a neuron, there's this amazing morphology of these cells, and I should find out where the pointer is, where you have a single cell body that we just heard about, and the neuron elaborates these beautiful dendritic and axonal processes. And so the question of how do you spatially and temporally regulate gene expression becomes much more complicated in neurons than it is in other cell types. And that's really clear when you look at a much simpler cell type like a lymphocyte. Not that lymphocytes are that simple, but in terms of getting a signal from the plasma membrane to the nucleus to regulate gene expression, altering function in the cytoplasm, and you look at a comparison here of a Purkinje neuron, and you think about getting a signal from a distal site all the way back to the cell body and into the nucleus, and then you think about being able to change the structure and function, the proteome of specific synapses or neighborhoods of synapses within that cell. It really raises questions about what are the underlying mechanisms that allow that sort of compartmentalization of activity-dependent gene expression. So, of course, when I talk about gene expression is much more complex than just activity-dependent transcription or translation, and it includes many different steps. And in my lab, we really focus on the step of transporting the signal from a stimulated synapse to the nucleus and then regulating the transcription of DNA to RNA. And then we focus to try to understand how you get that compartmentalized change in the proteome. We focus on localizing RNAs to specific sites within the neuron, on regulating their translation, on regulating the stability of the RNA, and also on regulating the stability or the degradation of proteins locally. But today I'm just going to tell you about this first part about the transport of signals from distal synapses to the nucleus and their function in regulating transcription in an activity-dependent manner. And we know that neurons are really specialized for fast electrochemical signaling. Even, I learned now, C. elegans neurons have rapid electrochemical signaling where you get stimulation throughout the neuron. But my lab is really interested in this active transport of signaling molecules from specific stimulated synapses through the process back to the nucleus where it can turn on gene expression. And I really became interested in that process when I was a postdoc in Eric's lab working in this system of a bifurcated aplesia sensory neuron contacting two motor neurons and stimulating one of the branches, and did a lot of work to show with Eric and colleagues in the lab to show that that depended on the localization and translation of RNAs that were present at that synapse, but also found that it required the transport of signals from the synapse back to the sensory neuron nucleus, things like ERK1 MAP kinase. And so when I started my lab, I wanted to understand how that could happen, how you could get that long-distance signaling. And one of the molecules that To-Yen Ching, who was a postdoc and now has his own lab in Singapore, looked at, he started just looking at candidate molecules, looking for transcription factors that were localized in the cytoplasm, but underwent regulated nucleocytoplasmic trafficking to impact transcription. And so one of them that he looked at is, one of the transcriptional regulators he looked at is called CRTC, or Krebs-regulated transcriptional coactivator 1. And he decided to look at it because of work from Mark Montminy and Mark Lebau's lab in non-neuronal cells showing that CRTC1 localized in the cytoplasm where it was phosphorylated and bound to 14:3:3 anchoring proteins. But when you had a coincidence of increase in intracellular calcium and intracellular cyclic AMP, that activated the phosphatase calcineurin, dephosphorylated CRTC, it inactivated Syk kinase so that it remained dephosphorylated, traveled to the nucleus, bound Krebs, and very potently, about 1,500 100-fold induced the transcription of Krebs-dependent genes, and we knew from the work of Eric and many others that Krebs was important for that transition from short to long-term memory. So we wanted to look at CRTC, and I just want to add that over the last year, we've been collaborating with Sébastien Curie and Bertrand Isadore at Nantes, who have identified 2 patients that have CRTC mutations who have the intellectual disability, so that helps guide some of the work that we're doing now. Toyen first asked, well, where is CRTC localized in neurons? And looking in hippocampal slices, if he looks here, if he looked at the CA1 region at higher magnification, he could see that CRTC in the basal state was present in the dendritic region in stratum radiatum. It was not present in the CA1 nuclei. And if he induced LTP using a chemical LTP protocol that activates every synapse, he get this very robust translocation of CRTC1 into the nucleus and disappearance from the dendrites. And if you look at in dissociated cultures where you can get higher resolution of the localization, we could see that CRTC1 localized in the postsynaptic compartments and in the dendrites of hippocampal neurons in culture. To really then ask, well, can we visualize the translocation from a distal site? Does it actually travel from a distal site back to the cell body and into the nucleus? Toyen expressed CRTC fused to a photoconvertible fluorescent protein, Dendra2, that normally fluoresces green. Brief UV light, it turns to red. He expressed this in hippocampal neurons. And then he would bathe them in caged glutamate and use a brief UV light that would both photoactivate the CRTC1 dendra and uncage the glutamate, and then ask if he could detect translocation back into the cell body and into the nucleus. And doing this kinds of experiments before photoactivation, it's all green, there's no red. You go at about 200 microns, photoactivate from green so that you get red now distally, and then go back to the cell body and look at optical sections through the cell body. And at 0 time point, there's nothing in the nucleus, but after about 10 minutes, you begin to see both the green and the red that translocated, so that actually had to come from a distal site. So using that type of a protocol, Toyen did a number of experiments where he would uncage and then test different mechanisms to see what was required for that translocation back to the nucleus. And so in this experiment, for example, where he mock uncages glutamate, you see that translocation into the nucleus that's consistent with fast external transport of about a micron a second. And if he mock uncages in the blue, it doesn't really translocate, and if you block AMPA and NMDA receptors, you don't get any translocation. And so using that paradigm, he was able to show that that translocation required NMDA receptor, L-type voltage-gated calcium channels, and very local calcium influx, so it was blocked by fast but not slow calcium chelators. It required active transport along microtubules. It required the dynein motor protein. And it was dependent on a nuclear localization signal that he identified, shown here with these basic amino acids. One of the things that he noticed when he was running one-dimensional immunoblots is that the protein ran as a big schmear in basal state. If you silence neurons with tetrodotoxin, it was a higher molecular weight. If you stimulated with bicuculin, it dropped. And if you treated the lysate with a calf intestinal phosphatase to remove the phosphates, it dropped quite dramatically in molecular weight. And when he looked at the primary sequence, he noticed it was highly enriched for serines and threonines and tyrosines, and ran two-dimensional gels and showed that if you look here in the green, that CRTC in silenced neurons runs at a high molecular weight towards the acidic isoelectric point consistent with being phosphorylated. But if he stimulated here with bicuculin, he would get a drop in molecular weight with all these different species of largely differentially phosphorylated CRTC1, which he could show if he phosphatase treats, it drops way down, and the remaining parts are due to acetylation of CRTC1. Martina De Salvo, a graduate student in the lab, went on to work in N2A cells to do mass spec to identify all the phosphorylation sites, and identified 50 different phosphorylation sites that were really highly conserved between aplesia, humans, mice. And these are all shown in different colors here. And we've gone on, this is really too many for us to be able to analyze systematically, but did go on to show Martina and Toyen that if you mutate 3 of these really highly conserved sites at serine 64, 151, and 245, that's what's responsible for dissociating CRTC1 from 1433 at the postsynaptic compartment and driving its transport into the nucleus. But what we've really been interested in over the past few years is the idea that perhaps different forms, different patterns of dephosphorylation could lead to sort of encode those different types of different synaptic stimuli and couple them to different patterns, programs of gene expression, so that this could really be a way that the phosphorylation pattern could encode stimuli to produce distinct patterns of gene expression. And consistent with that, Jennifer Akiro in the lab has shown that in hippocampal slices, if she induces either long-term potentiation or long-term depression using either tetanic stimuli, theta burst stimuli, or low-frequency stimuli, all of these stimuli will induce translocation of CRTC1 into the nucleus. And more recently, in experiments in which we've been interested in how catecholamines impact the formation of long-term memories and the activity-dependent transcription in CA1 neurons, she's also shown that optogenetic stimulation of locus coeruleus fibers will drive CRTC into the nucleus of CA1 neurons. Both LTP and LTD induction will trigger dephosphorylation of CRTC1. So you can see that if you look just at the total CRTC1, you can see that drop in molecular weight both with the LTD and LTP. And if we use a phosphospecific antibodies for one of those residues that's required for the activity-dependent nuclear import, we also see loss of signal at that particular phosphorylation at that site. So over the last year, a graduate student, Siobhan Bonanno, and postdoc Jennifer Akiro have undertaken really massive ChIP-seq experiments in LTD and LTP. I'm only showing you a couple of the tracks here. I was hoping I would have the complete analysis, and we're really close, but we don't quite have it yet. But the goal was to really try to see if we could understand whether with different patterns of stimulation we get different patterns of binding to DNA. And the quick answer is that there's an enormous amount of overlap, but there are also some specific sites of binding during theta burst LTP and low frequency LTD. And in all cases, however, the motif that it binds to, and that's about 65% of the time, is the CRE element. However, we do find it binding at other ChIP peaks at sites for other BZIP transcription factors, and we're doing biochemistry to look at interactions between CRTC1 and those other transcription factors. The other feature that Toyen, actually Jennifer, noticed when she was looking carefully at the sequence of CRTC1, and we actually did this because we were trying to develop antibodies against phosphorylated forms of CRTC1, and so we collaborated with David Eisenberg to try to model the structure of CRTC1 to see if we could determine what was going to be externally exposed, and so convenient for making antibodies is that this has a very large low complexity domain, so a domain that has a poor, sparse number of amino acids that are included in it, and that looks like the kind of a protein that would form these liquid-liquid phase separations, so the ability of proteins with low complexity domains or disordered regions to form interactions with one another that essentially form a compartment within the cell that's not surrounded by a membrane. And this is from a paper from Bob Tejan showing that one of the features that happens with these many transcription factors that have these low complexity domains is that they form hubs where they will combine other transcription factors and really form a hub for transcriptional regulation. And so we started to use a super resolution microscopy looking at CRTC1 and found that indeed if we stimulate CRTC1, we get the formation of these puncta in the nucleus, but also in the cytoplasm. They're dissolved by 1,6-hexanediol, which is seen with other low complexity domain aggregates. And Daniel Wetherill, a postdoc in the lab, has recently developed software to be able to automatically analyze these puncta and has found that you get an increase in the number, a significant increase in the number of puncta when you stimulate, but that's completely blocked when you prevent the dephosphorylation, so when you use cyclosporin A to block calcineurins. So this is consistent with the idea that dephosphorylation drives CRTC1 into these liquid-liquid phase separation compartments where it may be able to interact with other transcriptional regulators and drive a pattern of gene expression. And we're currently working to identify what other molecules are in that complex. So I want to just finish by taking sort of a step back to a new set of experiments that a really wonderful graduate student in my lab, Wendy Herbst, has been undertaking, where she really set out to, she wanted to identify additional novel synaptonuclear signaling molecules. And she spent a lot of time doing biochemistry, trying to isolate cell bodies and nuclei and processes, and failed at every corner, and then started using proximity ligation assays where she expressed a construct that had the exogenously expressed peroxidase apex-2 fused to GFP and surrounded by nuclear localization signals. So the idea was that she would express this in neurons, make a virus to express it. It would localize to the nucleus and then biotinylate proteins that were in close proximity to it. And then we could isolate the cells, and all of the biotinylated proteins would be present just in the nucleus, and we could then use TMT mass spec to identify nuclear proteins. And so after tinkering around, she was able to create a construct that really showed very beautiful localization to the nucleus. So you can see here's the apex-2 GFP with the NLS on both sides in the nucleus. It only will biotinylate. You can only see that signal when you add biotin phenol and hydrogen peroxide. And so you can see that it's really quite beautifully specific. And then she did experiments in which she transduced neurons with this virus-expressing apex-2 NLS. She actually included the cyclohexamide to block translation because she really just wanted to detect any proteins that went into the nucleus after stimulation. She then either silenced or stimulated the neurons with TTX or with bicuculin, then labeled with biotin and with hydrogen peroxide. And doing these experiments, first of all, this was a really beautiful way for us to begin to define the nuclear proteome of hippocampal neurons. So we were able to identify over 3,000 proteins that were very highly enriched for nuclear proteins. In the bicuculin stimulated neurons, we identified 51 proteins that were enriched in the nucleus by proximity labeling after bicuculin. The highest was CRTC1, which was somewhat satisfying because it told us that we were, by picking a candidate, we'd actually picked something that was important. We also identified 34 that were enriched in the TTX silenced neurons. This included, we actually didn't know this literature, but it included HDACs 4, 5, and 9, which have been shown to undergo nucleocytoplasmic trafficking with enrichment after silencing. And we've, in this past couple of months, Wendy has confirmed several of these, and I'm most excited about the ones that she's actually looking at in the TTX enriched. And I think that it's really opening up a new field for trying to understand issues about how the cell body and the processes interact with one another. So just to summarize, I've told you about one synaptonuclear signaling molecule, CRTC1, that localizes at the synapse where it's highly phosphorylated. It gets dephosphorylated. It travels back that long distance on microtubules in a dynein-dependent manner. It binds to specific sites in the DNA. We're just identifying what those are, leads to expression of different RNAs. It then gets rephosphorylated in the nucleus and leaves the nucleus. And that's actually one area that we're beginning to study because it turns out that catecholamines will block that rephosphorylation, and so will leave it active for a longer period of time in the nucleus. We're interested in understanding this complex regulation of CRTC1 phosphorylation and its role in liquid-liquid phase separation, and finally identifying novel synaptonuclear signaling molecules. And I wanted to just add a few points about, that are sort of broader than these particular studies. And that is, when I think about cell biology right now in neuroscience, I wanted to just say something about how much human genetics is guiding a lot of the work or contributing to a lot of the work that we're doing. And I pointed out these 2 mutations in CRTC1 that have now been identified in patients. And so having that when you're really to guide us as we try to determine what we're working on in other studies as well has been incredibly valuable, and I think for me incredibly satisfying in trying to go from being a very reductionist cell biologist to really thinking about how these processes of a molecule moving within a cell might actually contribute to brain functioning or dysfunction in a human being. And finally, as I think about, I've spent my career, at least as a neuroscientist, really thinking about how gene expression is regulated within neurons. I think about how what happens at the synapse, what happens in the dendrite, what happens in the axon, what happens in the cell body. And more as I read the work of my colleagues, I've begun to really think more about neighborhoods, local neighborhoods of synapses and glia and extracellular matrix. And the work that I think is still not really that rigorous, but incredibly intriguing about extracellular vesicles that may actually transport RNAs or of different types from one cell to another, I think also begins to break down those barriers that we think about as from the neuron doctrine of individual neurons in the brain. So I want to thank the people who've done the work. I think I've talked about several of them. Jennifer Akiro is a wonderful postdoc. Siobhan Bonanno just graduated 2 weeks ago. Wendy Herbst is a graduate student in the lab, and Daniel Wetherill is a new graduate student. I'm a new postdoc, and I want to acknowledge my collaborators. The work that I talked about today has been the work on CRTC1. Much of it has been a collaboration with Tom Odell, who is also a postdoc in Eric Kandel's lab right before I came, overlapping for a short period of time, and James Wolfschlegel, with whom we've collaborated on the mass spec, and finally the collaborators at Nantes for the CRTC human genetics, and I'm happy to answer any questions.
Fantastic. So we have time for some questions. We start here while the youngest crowd gets their courage up.
Kelsey, thanks very much for a really beautiful talk. You mentioned at the beginning the possibility or the observation of synapse-specific plasticity, and do you think CRTC1 plays a role in that, since it would mark the synapse by its absence or potentially by its presence in the nucleus?
Yeah, I mean, that's one of the attractive features of having a particular molecule that moves from the synapse to the nucleus, is that it actually provides some sort of spatial memory of where it came from. And it's something we're really interested in. It's challenging to identify what's different about those synapses, so it's difficult to get at it biochemically. We've been trying to do experiments. We've worked really hard to make a conditional knock-in that we could express CRTC at endogenous levels and visualize it and try to differentiate between synapses that have or don't have it. And we actually haven't been successful. We've just succeeded in the last year at making a conditional knockout, and that took a long time for a variety of different reasons. But it's a huge question. I think it's a really interesting question. Presumably there is something distinct about a synapse that has or doesn't have CRTC1.
Leslie?
So it's really fascinating that there are humans with CRTC1 mutations. Do you know what the lesion is? Is it like knocking out 4 of the serines that mediate the phosphorylation?
So they're both frame shift mutations, and they change the C-terminus, and the C-terminus is a transactivation domain. So you get full-length protein. So we've looked at that, and we're trying to see how it impacts, what it interacts with.
Frank and Stafford. I'll run.
Is the gene haploinsufficient in human?
Is what?
Is the gene haploinsufficient in human?
Oh, I should know that. I think it is, yeah.
You haven't told us anything about the phenotype in the mouse knockout then.
Oh, yeah. So the mouse knockout, there's a germline knockout, and that was easy to make, and other people have described it. And they're obese, so they become hyperphagic. It plays a really important role in the hypothalamus. The males are incredibly aggressive. They're very hard to breed. And so that's why we were trying to get a conditional knockout. It took us a long time to target the allele to flox it. We now have it, and we've crossed it with CAMK2 alpha Cre, and we don't get full knockdown. And we've crossed it with a number of other Cre lines, and we get 65, 70%. So now we're actually doing viral injections and getting knockdown. So the reason we want to do that is there are a number of labs that have looked at the phenotypes. There are learning phenotypes. There's a paper that shows that there's sort of depressive phenotypes in some of the models. But it's just there's a huge background of these other phenotypes.
Have you looked at all if these CRTC speckles you see in the cell, do they correspond to regions where target genes of CRTC assemble?
So that's what we're trying to do now. So I don't have the answer to that.
So thank you very much, Kelsey. Again, a round of applause. And now it is my great pleasure to call to the podium Professor Eric Kandel, our co-director, to deliver the final remarks. He has the toughest job. Thank you so much, Eric.
Thank you. It's a pleasure to follow Kelsey, I must say. One thing you may not be aware of, in addition to her academic accomplishments, she's the dean of a medical school. And to be able to carry on both of those activities simultaneously is not only remarkable, but it's absolutely characteristic of her. It is a privilege to know her and to have a chance to work with her. This has been a really superb day. One could not ask for more. Without exceptions, the talks have been superb. And we can thank Costa, Richard Axel. I don't know whether you've ever heard of him. He's a relatively unknown guy, but he's very bright. Carol Mason and Kelly Ramone for organizing this meeting. They've done a superb job. Since this has been the inaugural Mind-Brain Behavior Symposium, it implies that more symposia to come in the future. So let us hope that the future symposium meet the same high standards we've met today. We are living in an intellectually interesting time. For young people in the audience, I mean, people who really don't know the history of the field, people like Richard Axel, who are relatively new to it, let me put this into perspective. In the past, a few decades ago, mind, brain, and behavior were quite distinct areas of studies. It's only more recently, in the last couple of decades, that we've seen, as we've witnessed in the symposium, that mind, brain, and behavior have come together as a united discipline. Moreover, we have heard about the studies carried out in human beings as well as in experimental animals, using the animal studies to a richer understanding of the human brain. Our understanding of the mechanisms whereby processes mediated by the human brain is still limited. We therefore have much to look forward to in the years ahead, using animal studies, but also as we develop techniques for studying the human brain without in any way damaging it, we will be able to carry out experiments in humans that we haven't been able to do until recently. So I thank you all for coming and look forward to seeing you in the future. This is the first symposium. There'll be other symposia in the future. So I Thank you, and I wish you farewell and bon voyage.